Restriction enzymes are endonucleases which cut within the DNA strand, unlike exonucleases which cut the nucleotides from the ends. Restriction enzymes recognize and bind to target sites, which are 4 to 6 base pair sequences, commonly palindromic but not always. A palindromic sequence reads the same forward on one strand and backward on the complementary strand. Restriction enzymes cleave specifically within the recognition sequence or at a random site some distance away. Although the enzyme cleaves both strands of a double-stranded DNA, the enzyme is said by convention to make one cut in the DNA. Cleavage is often asymmetrical, generating sticky ends, or less frequently symmetrical, forming blunt ends. Sticky ends are small stretches of single-stranded DNA capable of self-ligation or ligation with a complementary region from another DNA molecule. The sticky ends possess 3' or 5' overhangs of 1 to 4 nucleotides. Blunt ends are universally compatible with other blunt-ended DNA and possesses a 5' phosphate group that promotes ligation. Blunt ends, however, are not as easily joined together as sticky ends. Besides the recognition sequence, restriction enzymes' ability to cleave is determined by the absence or presence of DNA modification. Many restriction enzymes only cleave unmodified DNA. Some are insensitive to modification and others only cleave modified DNA, such as methylated DNA. Without restriction enzymes' ability to cleave at specific sites, long chromosomal DNA strands would be inconvenient to work with. Thus, the scientists who first isolated and used restriction enzymes, Werner Aber, Hamilton Smith, and Daniel Nathans, were awarded the 1978 Nobel Prize for Medicine and Physiology. The enzymes these scientists isolated were from bacteria. Bacteria uses restriction enzymes as a defense mechanism, cutting the DNA of bacteriophages that attack them while protecting their bacterial DNA by modifications such as methylation. Since the first isolated restriction enzyme, hundreds of different restriction enzymes have been isolated. The restriction enzymes isolated have been named systematically. The first 3 letters are abbreviations of the bacterial species from which the enzyme has been isolated, and the 4th letter represents the strain. Roman numerals are used as part of the name when more than one enzyme has been isolated from the same species and strain. As an example of how the enzymes are named, take the enzyme EcoR5. This is the 5th restriction enzyme isolated from Escherichia coli strain RY13. Another example is the enzyme Hindi3, which is the 3rd restriction enzyme isolated from the genus Haemophilus, species Influenza, and serotype D. The hundreds of restriction enzymes have been classified into different types. Type 1 enzymes recognize asymmetric nucleotide sequences that comprise 2 components in any orientation, each about 4 base pairs, separated by a non-specific spacer of 6 to 8 base pairs. These endonucleases cleave non-specifically 1,000 base pairs downstream to the recognition site. Type 2 restriction endonucleases requires one recognition sequence and cleaves specifically on or close to the recognition sequence. Type 3 enzymes requires 2 inversely oriented recognition sites for cleavage, each about 5 to 6 bases and non-palindromic. Once bound to a recognition site, the enzyme slides along the DNA and upon encountering a second complex, cleaves downstream of the recognition site, typically 25 to 28 bases away. Type 4 enzymes recognize modified, typically methylated DNA and cleave approximately 30 base pairs away from the recognition site non-specifically. Among these different types of restriction enzymes, type 2 with specific cleavage sites have been the most widely applied in biotechnology. Those restriction enzymes that cut specifically produce equal numbers of fragments of specific sizes from complete digestion of the same genome or chromosome. The average size of fragments produced, or the average distance between restriction sites, depends on the number of base pairs in a restriction enzyme recognition sequence. For example, the probability that a 6-base recognition site will be found in the genome, based on the 4 bases in DNA, is equal to 1 over 4 to the power of 6, which is 1 over 4096. In other words, the restriction enzyme cuts on average once every 4096 base pairs. So if the genome size is 40,960 base pairs, how many fragments will there be on average? If you answered 10, you would be correct. Knowing the number of fragments will be useful in, say, creating a genomic library, as you could guess the minimum number of vectors you would need to have every fragment of the genome represented in the library. Another factor that may influence the number of fragments would be the timing of exposure of the DNA to a restriction enzyme, which determines if digestion is complete or partial. A partial digest is a restriction digest that has not been allowed to go to completion and thus contains pieces of DNA with some restriction endonuclease sites that have not yet been cleaved. On the other hand, a complete digestion has all copies of the DNA present cleaved at all the restriction sites of the enzyme used for digestion. The number of fragments also depends on whether the DNA is linear or circular. If a linear DNA molecule having one recognition site for a restriction enzyme is digested, 2 fragments would be produced. However, if a closed circular DNA molecule with a recognition site for an enzyme is digested, it would change the circular DNA into a single linear DNA fragment. After the discovery and isolation of restriction enzymes which could cut DNA, it was demonstrated that these enzymes could be used in the construction of genetic maps. Here is an example how a genetic map is constructed. DNA saved from a cloned DNA fragment is used to prepare 2 single digests and a double digest. Then the 3 digests are electrophoresed to show the fragments obtained in each digest on a gel. The restriction sites are located on the DNA by first comparing the double digest to one of the single digests. Take, for example, BAMH1. Since both digests have the 3000 base pair fragment, it must be the 2500 base pair fragment in the BAMH1 digest that BGL2 has cut in the double digest to produce pieces of 1000 base pairs and 1500 base pairs. But there are 2 possibilities. The 1500 base pair fragment or the 1000 base pair fragment could be located at the center. So now a comparison is made between the double digest and the BGL2 digested fragment. Since there is a 5000 base pair fragment in the BGL2 digested DNA, 2 fragments in the double digest must add up to 5000 base pairs. Thus, the 1500 base pair fragment must be at the center and the map is as shown here. Since the initial use in mapping, restriction enzymes have been used for many other purposes. Sequencing is carried out on DNA cut into short fragments as DNA polymerase, which extends the primer, may not retain its activity when incubation is prolonged. Recombinant DNA technology uses DNA cut to a short fragment that is suitable to be inserted into a vector, and the vector is also cut, but not so many times as to make it difficult to put it together again. DNA profiling and diagnostics uses specifically cleaved DNA so that the results can be replicated. And the list of restriction enzyme uses goes on, showing how important restriction enzymes are in biotechnology. To summarize the ideas on restriction enzymes covered in this video, 1. Restriction enzymes are endonucleases that recognize and bind to target sites, which are 4 to 6 base pair sequences, commonly palindromic but not always. 2. Depending on the restriction enzyme, cleavage may be specifically within the recognition sequence or possibly at a random site some distance away, symmetrical or asymmetrical, and affected or not by DNA modification. 3. The number of fragments formed on cleavage by an enzyme is determined by the number of bases in the recognition sequence, the size of the DNA being fragmented, whether DNA is linear or circular, and whether digestion is complete. 4. Restriction sites of enzymes act as markers on a chromosome in restriction mapping. 5. Restriction enzymes are a molecular tool in many biotechnology procedures. Now to review the concepts covered with a few questions. Question 1. Restriction enzymes are such a useful molecular tools because A. They cut up bacteria that invade and contaminate sterile cell cultures. B. They cut DNA at defined recognition sequences, and the longer the recognition sequence, the more often it occurs in any given sample of DNA. C. They make molecular biology more exciting because you never know where DNA sequences will be cut. D. They cut DNA at defined recognition sequences, and the shorter the recognition sequence, the more often it occurs in any given sample of DNA. The correct answer is D. Answer A is incorrect, as bacteria use restriction enzymes to cut up bacteriophage or viral DNA. Answer B is incorrect, as the longer the recognition sequence, the harder it would be to find that sequence in the sample DNA, hence the longer the fragments. Answer C is incorrect, as the restriction enzymes that cut at specific sequences are the most useful as results can be replicated. Question 2. Genetic engineers use restriction enzymes to cut DNA into smaller lengths. The recognition sequences of several restriction enzymes are shown in the table. The asterisk symbol denotes the restriction site or position of the cut. Now consider a length of double-stranded DNA with the sequence shown. Which enzyme or enzymes will cut this piece of DNA? A. EcoR1, B. Hindi3, C. ALU1 and Hindi3, D. ALU1, Hindi3, and HAE3. The answer to this question is C, as only the recognition sequences of ALU1 and Hindi3 enzymes can be found in the sequence given as shown. Question 3. Which of the following is true about the restriction enzyme BAMH1, which cuts the DNA after the 5' guanosine on each strand? A. When it cuts DNA, it creates a 5' overhang. B. When it cuts DNA, it creates a 3' overhang. C. It is classed as an exonuclease. D. It can cut both single and double-stranded DNA. The answer is A. It creates a 5' overhang as shown in the diagram. Question 4. A plasmid with restriction sites for BAMH1 and EcoR1 is shown. Several restriction digests were performed using these two enzymes, either alone or in combination. The digests were electrophorused and the fragments were observed as shown. Which lane shows the fragments produced when the plasmid was incubated with EcoR1 and BAMH1? A1, B2, C3, D4, E5. The answer is D. There has to be 4 fragments of size 2, 4, 6, and 8 kilobases. Question 5. A bacterial plasmid is digested with BAMH1, XHO1, and a combination of BAMH1 plus XHO1. These 3 reaction mixtures are then electrophorused on an agarose gel, resulting in the following restriction fragments. BAMH1 shows a single 4 kilobase band, XHO1 shows 2 bands of 2.8 kilobases and 1.2 kilobases. BAMH1 plus XHO1 shows 3 bands of 1.7 kilobases, 1.2 kilobases, and 1 kilobase. Draw a map of the plasmid, including the restriction sites and the distance between them. To draw the map, let's first analyze the information obtained from the gel. It can be seen that BAMH1 cuts once, as there is only one fragment, and XHO1 cuts twice, as there are 2 fragments. Comparing the double digest to the XHO1 digest, the fragment of size 1.2 kilobases is not cut by BAMH1. So BAMH1 cuts the 2.8 kilobase fragment into 2 fragments of 1.7 and 1.1 kilobases. The map is shown here. So that's a summary on restriction enzymes. If the video helped you know more about the restriction enzymes, please give it a thumbs up and subscribe to support the channel. Thank you.